Hello there, and welcome to The Sleepy Scientist. I'm so glad you've decided to spend a little time winding down with me. Whether you're snuggled up under the covers, staring softly at the ceiling, or letting your eyes slowly close while your mind floats, tonight's gentle exploration is here to soothe, to fascinate, and hopefully, to lull you towards sleep. Now, a small warning. This video might be a touch too interesting. If you find yourself wide-eyed at midnight contemplating genetic codes and molecular dances, well, at least you'll be better informed at 3 in the morning. There are worse things to ponder in the dark than the poetry of your own DNA. Before we curl up inside the double helix, I'd love to ask a tiny favour. If you enjoy this quiet journey tonight, a like and a subscribe go a long way in helping me make more of these relaxing deep dives. And while you're here, why not let me know where in the world you're watching from? Maybe your city is just beginning to yawn into night, or perhaps you're halfway through your midnight tea. I'd love to hear what time it is by you and where this sleepy science finds you tonight. Alright, get comfortable, breathe in gently, and let's drift into the beautiful hidden story written inside you, the quiet script of life, your DNA. And just before we begin, a gentle reminder. This video is for educational and relaxation purposes only. While we'll be talking about genetics and health-related topics, nothing here should be taken as medical advice. If you have questions about your health or genetics, it's always best to speak with a qualified medical professional. Inside every one of your cells, there lives a tiny storyteller, a molecule that's been writing your personal tale since the moment you were just a single cell. This storyteller has no ink, no pen, and no paper. It uses only 4 letters, just 4. And with them, it weaves everything from the way your eyes catch the light to how you process caffeine on a Monday morning. Those 4 letters, A, T, C, and G, stand for the chemical bases adenine, thymine, cytosine, and guanine. Together, they form the entire alphabet of your DNA, or deoxyribonucleic acid, which is just a fancy way of saying the twisty stuff that makes you, well, you. It might sound limiting at first, there are only 4 letters to describe an entire human being. But imagine this. With just 26 letters, we've written entire libraries, symphonies, grocery lists, and the complete works of Shakespeare. So when nature writes your biological novel with 4, it's not so much a constraint as it is a miracle of efficiency. These letters pair up in very specific ways. Adenine always pairs with thymine, and cytosine always pairs with guanine. It's a bit like an old-fashioned dance where everyone has their one perfect partner. No cutting in allowed. They line up in pairs, forming a long, twisty ladder that coils into the famous double helix, DNA's iconic shape. Now, to understand just how much story your DNA holds, let's take a moment to picture the scale. In each of your cells there are about 3 billion base pairs, 3 billion steps on that molecular ladder. If you were to stretch the DNA from a single human cell out flat, it would be over 2 meters long. That's taller than most refrigerators, and definitely longer than most yoga mats. And yet somehow it's all tucked neatly into a nucleus so small you'd need a microscope to even find it. It's like folding a long poetic scroll into a grain of sand, except the scroll can make enzymes, repair wounds, digest pizza, and sometimes give you dimples. Every combination of A, T, C, and G makes up the instructions for something, perhaps the production of a certain protein, or a blueprint for how your body should grow a fingernail, form a freckle, or respond to pollen. Think of these combinations as little words or sentences. Some are short and sweet, others go on like a rambling family recipe. Together they make up genes, and your genes are the chapters in the grand book of you. Now here's a curious thing. All humans share about 99.9% of their DNA. So you, me, and the person delivering your mail are almost genetically identical. It's in that tiny 0.1% where the quirks live. The difference between having curly hair or straight, whether your earlobes are attached or free-floating, and yes, even whether you love or despise the taste of coriander. And DNA isn't just inside humans, of course. It's the universal script of life. Every living thing that grows, reproduces, or wiggles has dentucts somewhere inside it. Trees, jellyfish, mushrooms, mosquitoes. Everyone's reading from the same basic code, just with a few editorial changes. You even share about 60% of your DNA with a banana. That doesn't mean you're half fruit, of course, but it does hint at the deeply shared history of all living organisms on Earth. So how does DNA actually do anything? How does a simple code become a person or a tree or a hummingbird? It starts with the order of those letters, the sequence. If DNA is the alphabet, then the sequence is the story being told. Rearranging the letters changes the instructions. A single letter swap can sometimes have profound effects, like changing the function of a protein or stopping one from forming altogether. To make use of its information, DNA doesn't act alone. It needs help. It's the mastermind in the back room, whispering instructions to molecules like RNA, which ferries messages out into the cell, and proteins, which do most of the actual heavy lifting, building things, breaking things down, regulating, repairing, and generally keeping the lights on. But we'll meet those helpful molecules another time. For now, just know that DNA is a bit like a quiet librarian who never leaves their archive. It stores the instructions for life, it passes those instructions along with incredible precision, and sometimes, just sometimes, it introduces a little change. Not because it wants to stir up drama, but because copying 3 billion letters perfectly every time is, well, hard. Those changes, or mutations, can be good, bad, or completely silent. Sometimes they do nothing at all, sometimes they cause illness, and sometimes they turn wolves into poodles. Evolution loves to tinker, and DNA is the canvas. And though it might sound like DNA is a cold clinical code, it's anything but. It's deeply personal. Your DNA carries echoes of your ancestors, thousands of years of survival and migration wrapped into your cells. It quietly remembers the storms they weathered, the climates they adapted to, the foods they hunted or grew. It even remembers ancient cousins we once shared the earth with, Neanderthals and Denisovans, whose genes still whisper inside many of us today. So tonight, as you drift towards sleep, think of the A's and T's, the C's and G's, all dancing together quietly in the dark. Writing and rewriting the tiny poem of your existence, every second in every cell. A poem you never see but that you carry with you always. And isn't that just a little bit magical? Picture a spiral staircase, one that stretches not upward toward a lofty ceiling but inward, coiling tightly into the core of every cell in your body. This isn't an ordinary staircase, of course. It's made of molecules, winding like a slow-motion whirlpool, forming what is perhaps the most iconic structure in all of biology, the DNA double helix. That elegant twist wasn't always known to us. For centuries, people had no idea how traits were passed on. Farmers knew how to breed crops and animals with desirable features, and families observed that blue eyes or a distinctive nose often showed up generation after generation, but no one knew why. There were clues, certainly. By the 1800s, scientists had noticed that cells carried mysterious thread-like structures, chromosomes, that seemed to divide and reappear with curious precision, but what exactly was inside them remained a mystery. Then came the year 1953. A pair of researchers, James Watson and Francis Crick, were working at the Cavendish Laboratory in Cambridge, puzzling over how genetic material might be structured. They had some ideas, but they were missing a crucial piece of the puzzle, literally. That piece came from Rosalind Franklin, a brilliant X-ray crystallographer whose images of DNA revealed a ghostly cross-shaped pattern. One of her photos, known as Photo 51, captured DNA in its helical glory. Watson and Crick, using her data, though controversially and without her permission at the time, built a model that showed DNA as a double helix. Two strands twisting around each other like a zipper in 3 dimensions. When they saw it, everything clicked. The base pairing rules, the stability, the way information could be copied, it all made sense. The double helix wasn't just beautiful, it was functional. Now, let's look a little closer at this twisty marvel. Imagine a rope ladder, but instead of wooden rungs and nylon ropes, you have alternating sugar and phosphate groups forming the sides, and base pairs, adenine with thymine, cytosine with guanine, forming the rungs. This whole structure coils gently into a right-handed spiral, making one full turn every 10 base pairs or so. The sugars are 5-carbon molecules known as deoxyribose, and they were attached to phosphate groups which link the sugars together like the beams of a scaffold. This sugar-phosphate backbone runs in opposite directions on each side, what scientists call anti-parallel strands. It's a bit like two escalators next to each other, one going up and one going down, except both are part of the same molecule. The rungs of the ladder, the base pairs, are held together by hydrogen bonds. These are not strong bonds individually, but there are so many of them that together they make DNA quite stable. Adenine pairs with thymine using 2 hydrogen bonds, while cytosine pairs with guanine using 3. That extra bond gives the CG pairing a little more strength, and as a result, regions of DNA with more CG pairs tend to be more tightly wound. But the magic of the double helix isn't just in how it's built, it's in how it stores and copies information. Because of the base pairing rules, each strand of the helix contains all the information needed to reconstruct the other. If you have a strand that reads ATGCA, the opposite strand will read TACGT, like mirror images whispering secrets across a molecular bridge. This means that when a cell divides, it can unzip the DNA and use each strand as a template to build a new one, ensuring that each new cell gets an identical copy of your genetic instructions. The helical shape also allows the DNA to be incredibly compact. If the DNA from just one human cell was stretched out, it would be about 2 meters long. Multiply that by the roughly 37 trillion cells in your body, and you have enough DNA to stretch from here to the moon and back, many times over. Yet thanks to its spiral nature and some very clever folding mechanisms, it all fits neatly inside microscopic nuclei. This coiling isn't just for space saving. The helical structure protects the genetic code from damage and makes it easier for cellular machinery to read and copy the DNA when needed. Think of it like a scroll wound up tightly. When it's time to read a section, it can be carefully unrolled without disturbing the rest. Interestingly, the helical twist isn't uniform in all cases. Most DNA exists in what's called the B form, this classic right-handed helix that Watson and Crick described. But under certain conditions, DNA can adopt other shapes. There's the A form, which is a shorter and more tightly wound helix, and the Z form, which is left-handed and kinda squiggly, like DNA's rebellious cousin who listens to jazz and questions authority. Scientists still don't fully understand the role of these alternative forms, but they suspect they may be involved in gene regulation and other nuanced cellular behaviours. All of this, the shape, the bonds, the coiling, the twisting, is why DNA works so well. It is not just a storage device. It's an active, responsive, finely tuned molecular structure that has been refined over billions of years of evolution. It's strong enough to endure the constant churn of cell division, yet flexible enough to interact with enzymes and proteins that read, repair, and regulate it. And here's a quiet thought to rest on. The double helix is not just a metaphor for life's complexity. It is life's complexity, rendered in miniature. With its spirals and symmetry, its hidden messages and careful pairings, DNA doesn't just carry information, it sings it. It hums a quiet melody of who you are and who you might become. So the next time you curl your fingers or take a breath or fall gently into sleep, remember, within you, every cell is turning pages in a spiral book. A book where the story is written not in ink, but in atoms. Where the letters do not sit still, but twist and coil and shimmer. And all of it, every line and loop, begins with a twist. If you have ever tried to cram a duvet into a suitcase, you already have a small idea of what your cells are dealing with. DNA, as we've seen, is incredibly long, about 2 meters of it in every single human cell, and yet somehow it all fits neatly inside a tiny nucleus, a space so small you'd need a powerful microscope just to notice it. This isn't just impressive, it's essential. If your DNA weren't folded with exquisite precision, it would be tangled like last year's fairy lights and your cells would be in utter chaos. So how does nature manage to fold this molecular epic with such elegance? The answer lies in the art of packaging, a combination of structure, organisation, and frankly, a little molecular origami. And at the centre of this operation are proteins known as histones. Histones are like the spools in an old-fashioned sewing kit, around which your DNA winds itself. Each spool is made up of 8 histone proteins bundled together in what's called a histone octamer. DNA wraps around each of these little protein cores, completing about 1.5 loops before moving on to the next. This structure, DNA plus histone, is known as a nucleosome. Imagine beads on a string, where the string is your DNA and the beads are nucleosomes. These beads don't just sit in a line forever. They twist and fold again into tighter loops, forming fibres that can be condensed further still, depending on how busy that section of DNA is. If a part of your DNA needs to be read frequently, for example, the instructions for making insulin or digesting food, it tends to stay loosely packed. This open configuration is called euchromatin, and it's sort of like leaving a cookbook open on the kitchen counter. Easy to access, ready to use. But if a region doesn't need to be read often, or perhaps contains information that should not be read at all, it gets tightly bundled up in a form called heterochromatin. This is the day equivalent of shoving old files into a locked cabinet. Still there, still important, but not meant for casual browsing. Altogether, this careful organisation allows DNA to be both compact and accessible. When the time comes to copy or repair a section of DNA, or transcribe a gene into RNA, the chromatin, the combination of DNA and protein, can temporarily open up to allow enzymes in. Then it folds itself away again, neat as ever. Now, the entire collection of DNA inside a single human cell is known as the genome. And this genome is broken into manageable sections called chromosomes. You've probably seen the classic textbook image of those little X-shaped figures floating serenely in space. Those are chromosomes caught at their most photogenic, right before a cell divides. In reality, chromosomes don't look like X's most of the time. They're usually more like loose, noodly fibres packed into the nucleus like a well-organised drawer of socks. But when a cell prepares to divide, it duplicates its DNA and coils everything tightly to make sure the copies can be split evenly between the 2 new cells. That's when chromosomes take on their iconic symmetrical X shape. Each half of the X is a chromatid, essentially one copy of the DNA, and the 2 chromatids are joined at a central point called the centromere. Humans have 46 chromosomes in total, 23 pairs, with 1 set inherited from each parent. That includes 22 pairs of autosomes, non-sex chromosomes, and 1 pair of sex chromosomes. Most women have 2 X chromosomes, while most men have 1 X and 1 Y. These pairs carry all the instructions for making and maintaining a human body, plus a few surprises like genetic predispositions and ancient family secrets. It's worth noting that while every cell in your body, except red blood cells and a few specialised types, contains the same DNA, they don't all use it in the same way. Your skin cells and brain cells, for example, have identical genetic material but express different sets of genes. This is made possible by the way DNA is packaged and regulated. Some genes are silenced in certain cell types and activated in others, depending on what that cell's job is. It's a bit like every cell being given a complete library, but only reading the books that apply to its particular career path. A liver cell isn't especially interested in building neurons, so it shelves those chapters and highlights the ones about detoxifying the bloodstream. This selective reading is influenced not only by the arrangement of histones, but also by chemical tags added to the dig or the histones themselves. These tags form part of what's called the epigenome, a dynamic set of instructions layered on top of the genome, helping determine when and where genes are expressed. Think of it as the bookmarks, sticky notes, and highlighter marks of your genetic library. And because the packaging and tagging of DNA is so crucial, your cells have an entire crew of proteins dedicated to managing it. These include chromatin remodelers, enzymes that help unwind or tighten DNA as needed, as well as repair proteins, scaffolding proteins, and more. It's like a whole team of librarians, janitors, and security guards working night and day to keep everything in order. Occasionally things go awry. A mutation in the proteins that package or regulate DNA can lead to serious consequences. Improperly folded or misregulated DNA is linked to various diseases, including many cancers. When genes that should be silent are accidentally turned on or vice versa, cells can begin behaving in unpredictable ways. That's why understanding how DNA is packaged isn't just a question of biological curiosity, it's vital for medical science too. But most of the time, your cellular filing system works beautifully. Right now, as you listen to this, your DNA is winding and unwinding, opening and closing, turning the pages of your internal instruction manual with quiet efficiency. So as you begin to relax deeper into the evening, think of the microscopic world inside you, of the invisible librarians rolling out scrolls and gently shelving them again. Every breath you take, every beat of your heart is supported by this complex choreography of folding, organizing, and reading your DNA. It's an astonishing feat of natural engineering, a biological ballet that never ends, and yet it all unfolds in silence, in the dark, deep within each of your trillions of cells. Tidy, quiet, and profoundly elegant, just like your genome. If DNA is the grand library of life, then genes are the books sitting neatly on its endless shelves. Each one holds instructions for something specific, how to make a particular protein, when to grow a hair follicle, or how to process a spoonful of honey. But don't let the term gene fool you into thinking they're always long or complex. Some are surprisingly short, others are epic in scale, and yet together, these little stretches of code form the foundation for just about everything your body is and everything it does. The average human has somewhere between 20,000 and 25,000 genes, scattered across the 46 chromosomes in their cells. These genes make up only about 1 to 2% of your entire genome. That's right, over 98% of your DNA doesn't directly code for proteins. And for a long time, scientists referred to this non-coding DNA as junk. That term has since fallen out of favour because, as it turns out, much of this so-called junk is quite important. But we'll get to that. Let's focus first on the genes that do code for proteins, because those are the stars of tonight's quiet little story. Genes are made up of specific sequences of the 4 DNA bases: adenine, thymine, cytosine, and guanine. These sequences contain the instructions for building proteins, which are the molecular machines that carry out nearly every function in your body. From digesting food to healing cuts to making your eyes sparkle in the sunlight, proteins do the heavy lifting. Now, genes are not read all at once like flipping through a magazine. Instead, they're transcribed, a kind of copying process where the cell makes a working version of the gene using a molecule called RNA, or ribonucleic acid. You can think of RNA as the cell's messenger or scribe, making temporary notes so the original DNA can remain safely tucked away. Before transcription happens, though, the cell needs to locate the right gene. It does this using something called a promoter, a short region of DNA that acts like a start here sign. Proteins known as transcription factors bind to the promoter and help recruit an enzyme called RNA polymerase. This enzyme is the real workhorse. Once it's in place, it unzips the DNA and begins to copy the sequence letter by letter, creating a strand of messenger RNA. Now here's where things get a bit clever. The gene sequence includes regions called exons, which are the parts that actually code for proteins, and introns, which are non-coding segments scattered in between. After the RNA is made, the introns are snipped out and the exons are stitched together in a process called RNA splicing. The final polished messenger RNA is now ready to be translated into a protein. This whole system, coding regions, splicing, promoters, is like a recipe that includes helpful instructions, a few scribbled notes in the margins, and perhaps a paragraph you choose to skip because it's just someone rambling about the history of olive oil. The proteins produced from genes are built from chains of amino acids. There are 20 different amino acids, and the order in which they're assembled determines the protein's structure and function. Some proteins act as enzymes, speeding up chemical reactions. Others form structural components like collagen in your skin or keratin in your hair. Some are transporters like hemoglobin, which carries oxygen in your blood. And then there are the regulatory proteins. These are the ones that help control when and where genes are expressed. It's a bit like having editors and managers inside your cells, keeping everything running smoothly and ensuring the right proteins are made at the right time. One of the fascinating things about genes is how flexible they are. Through a process known as alternative splicing, a single gene can lead to the creation of multiple proteins. By rearranging which exons are included or excluded, cells can produce different versions of a protein from the same gene, each with its own specific role. This is part of why humans, with a relatively modest number of genes compared to some other organisms, can still produce an astonishing variety of proteins. For example, a humble water flea has more genes than you do. But you, dear listener, have complexity on your side. Your genes don't just sit around producing one protein each. They multitask. And remember that large percentage of your DNA that doesn't code for proteins? Well, some of it plays an important regulatory role, like enhancers and silencers, which help control how strongly a gene is expressed. Other segments might have structural roles, or they could be remnants of ancient viruses that once invaded your ancestors' cells. Yes, really, your DNA is peppered with the genetic fossils of long-extinct viruses. Most of them are harmless now, but some still whisper instructions that affect how your genes behave. It's also worth noting that genes don't operate in isolation. Many traits, like height, intelligence, or susceptibility to certain diseases, are influenced by the combined activity of many different genes, along with of environmental factors. That's why we say most traits are polygenic and complex, rather than simple and determined by a single gene. Remember the idea of a gene for something, like a gene for musical talent or a gene for being good at parallel parking? It's a bit misleading. Most traits aren't so cut and dry. There might be genes that influence aspects of a trait, but rarely is there one gene pulling all the strings. Life, it turns out, is more like a symphony than a solo. Still, there are genes that have strong, well-understood effects. For example, mutations in the BRCA1 and BRCA2 genes are linked to increased risk of breast and ovarian cancers. Mutations in the gene that produces hemoglobin can cause sickle cell anemia. These are powerful examples of how changes in even one gene can have far-reaching consequences. Yet most of your genes are quietly doing their jobs day in and day out, without drama or fuss. They keep your heart beating, your skin renewing, and your mind humming along. So as you rest tonight, imagine those tiny sections of DNA inside each of your cells, each one like a tiny chapter in your biological novel. Some chapters are long and detailed, others are short and sweet, and every one of them is necessary. Together, your genes form the delicate script of your body. A living manuscript, written in molecules, edited by evolution, and read by the machinery of life, one cell at a time. Imagine your DNA as the quiet architect, tucked away in the corner office of the cell, carefully guarding the blueprints of your body. It doesn't leave the office, doesn't get its hands dirty, but it writes all the instructions for how everything should be built. The ones doing the actual construction, the real builders, are proteins. And getting from DNA to protein is a two-step process known as gene expression. It's a calm, elegant assembly line, first transcription, then translation. Let's begin with transcription. This is where your cell makes a working copy of a gene using a molecule called messenger RNA, or mRNA for short. If DNA is your original reference book, beautiful but delicate, then mRNA is the dog-eared photocopy you can scribble on, fold into your pocket, or run through the rain with. It all starts when an enzyme called RNA polymerase binds to a section of DNA at the beginning of a gene. This area is known as the promoter, and you can think of it as a big neon sign that says "Start here." Once RNA polymerase locks on, it unzips the DNA just a little bit, enough to one strand, and begins building a complementary strand of mRNA, one letter at a time. Adenine pairs with uracil in RNA instead of thymine, so the letters shift slightly, but the message remains intact. This process unfolds smoothly and quietly. Like a scribe writing by candlelight, RNA polymerase transcribes the gene with care, crafting an mRNA molecule that carries the instructions out of the nucleus and into the bustling main floor of the cell, the cytoplasm. Now we move to the next phase, translation. This is where the cell reads the mRNA and actually builds the protein. For this task, it relies on a truly fascinating machine known as the ribosome. These tiny molecular factories float around in the cytoplasm or attach themselves to the rough endoplasmic reticulum, which is a name that sounds like a Harry Potter spell, but is actually just a folded membrane system inside the cell. The ribosome clamps onto the mRNA and begins to read it 3 letters at a time. Each 3-letter sequence is called a codon, and each codon corresponds to a specific amino acid, the building blocks of proteins. There are 20 different amino acids, and the order in which they're strung together determines what kind of protein is made. To help with this delicate work, the ribosome calls in a helper, transfer RNA or tRNA. Each tRNA carries a specific amino acid and has an anticodon that matches up with the codon on the mRNA. It's a bit like a puzzle piece clicking into place. As each codon is read, the appropriate amino acid is added to the growing chain one by one, in perfect order. Over time, usually just a few minutes, a long, ribbon-like chain of amino acids emerges from the ribosome. Once the full sequence is built, this chain will begin to fold into a precise 3-dimensional shape. That shape determines the protein's function. Some fold into globular blobs, others into long fibrous strands. The tiniest changes in folding can make a big difference. A misfolded protein can lead to diseases like cystic fibrosis or Alzheimer's. But most of the time, the folding happens beautifully, like origami performed in silence. Some proteins stay right where they're made, working inside the cell that built them. Others get shipped off to different parts of the body. A protein made in a skin cell might help hold neighbouring cells together. One made in a pancreatic cell might be insulin, released into the bloodstream to regulate sugar. Still others become enzymes, speeding up chemical reactions, breaking down food, repairing DNA, or building new molecules. It's also worth noting that this whole system is staggeringly efficient. A single cell can make thousands of proteins every second, and each one is built with exquisite accuracy. Occasionally mistakes happen, of course, but the cell has proofreading systems and quality control checks in place. It's like a molecular publishing house where typos are rare and editors are always on call. Now, what tells a cell which proteins to make and when? That's the subtle dance of regulation. Not every gene is active in every cell, some are turned off entirely, others are only turned on under certain conditions, when you're hungry for instance, or fighting off an infection. Special molecules called transcription factors bind to DNA and help control which genes are expressed and at what level. Environmental signals like light, temperature and stress can also influence gene expression, which is part of how your body adapts to different situations. Take, for example, the gene that helps you digest lactose, the sugar in milk. In most people around the world, this gene is active during childhood and then gradually shuts down. But in some populations, due to a relatively recent evolutionary twist, the gene remains active into adulthood. That's why some people can enjoy a tall glass of milk without incident, while others might deeply regret the experience. Even things like exercise or emotional stress can change gene expression in real time, not by altering the DNA itself, but by adjusting how the information in your genes is used. It's a bit like turning the volume up or down on different channels depending on what your body needs in the moment. This ability to respond, to shift and adapt is part of what makes the DNA to protein process so remarkable. It's not just a fixed script. It's a dynamic performance, responsive to your life, your choices, even your thoughts and feelings to a surprising degree. And here's something quietly astonishing. You're made of proteins. Not just built by them, made of them. Your skin is up here, your muscles, your blood, your brain, even the keratin in your eyelashes and the enzymes in your tears. All are crafted from proteins, carefully assembled and folded according to instructions that were whispered out of your DNA, read by RNA, built by ribosomes and folded with love. So as you drift into rest tonight, imagine that behind the scene, your cells are still gently humming with activity. Messenger RNA is still being written. Ribosomes are still clicking into place. Amino acids are still lining up like dancers waiting for their cue. All orchestrated by a molecule so small you've never seen it, yet so essential it writes the very rhythm of your life. And through it all, there's no shouting, no chaos, just quiet precision. A whisper of instructions passed from one molecule to the next, a peaceful production line that never sleeps, just like your body gently working even now as you begin to let go of the day. Even the most careful scribes make mistakes. A letter swapped here, a line skipped there. In the vast library of your DNA, with over 3 billion base pairs written into every cell, occasional typos are almost inevitable. These little changes in the genetic code are called mutations, and they're one of nature's most fascinating and unpredictable editing tools. Sometimes they're harmless, sometimes they're helpful, and sometimes they cause trouble. But without them, evolution would grind to a halt, and the diversity of life we see today would be reduced to one very bland version of biology. Let's start with the basics. A mutation is simply a change in the DNA sequence. It might involve a single letter being swapped, say an adenine replaced by a cytosine, or an entire section being deleted, duplicated, or moved to a new location. Most of the time, these changes happen during DNA replication, when a cell copies its genetic material to prepare for division. Given the speed and complexity of this task, it's impressive how accurate it usually is. Your cells have proofreading mechanisms, error-checking enzymes, and molecular correction fluid, so to speak. But even the best editors miss a spot now and then. There are several kinds of mutations. The simplest is a point mutation, where just one base pair is altered. This might seem trivial, but in certain contexts it can make a big difference. For example, sickle cell anemia, a serious blood disorder, is caused by a single point mutation in the gene for hemoglobin. Just one letter changed, and suddenly the red blood cells take on a crescent shape instead of their usual round form. That tiny shift affects how blood flows, how oxygen is carried, and how the body responds to physical stress. Other mutations might involve insertions or deletions, adding or removing a few letters in the DNA sequence. If the number of letters added or removed isn't a multiple of 3, this can cause what's called a frame shift mutation. Since the genetic code is read in 3-letter segments, remember codons? Shifting the frame throws off the entire reading process, like moving the starting point in a sentence. For instance, the cat sat becomes hecate sat. Suddenly the message becomes garbled. Then there are copy number variations, where large sections of DNA are duplicated or missing entirely, and chromosomal mutations, where whole chunks of chromosomes are swapped or inverted. These larger changes can lead to conditions like Down syndrome, which is caused by an extra copy of chromosome 21, or certain types of cancer, which often involve complex rearrangements in the genome. But here's the twist. Mutations aren't always bad. In fact, they're often neutral. Your DNA contains vast regions that don't code for proteins, and a change in these areas might have no effect at all. Think of it as correcting a typo in a footnote of an unpublished manuscript. No harm done. Sometimes mutations can even be beneficial. Imagine a mutation that allows someone to digest a new food, resist a disease, or tolerate a harsh climate. Over generations, such advantageous mutations can spread through a population, gradually shaping the traits of entire species. This is how evolution works. Tiny changes, selected by nature, accumulating over time. Take, for instance, the gene that allows adults to digest lactose, the sugar found in milk. Most mammals lose the ability to break down lactose after weaning, but certain human populations, particularly in Europe and parts of Africa, evolved a mutation that keeps the lactase gene active into adulthood. This single change meant those people could benefit from milk as a food source, especially during times of scarcity. It was a dietary superpower passed on through generations. Or consider resistance to malaria. People with one copy of the sickle cell mutation have a reduced risk of developing severe malaria, offering a strange sort of genetic advantage in regions where the disease is common. The mutation is dangerous in its double form, but protective in a single copy, a biological balancing act shaped by environmental pressures. And then there are traits we don't fully understand yet. Why do some people need only 4 hours of sleep while others need 9? Why do some people feel intense fear at the sight of a spider, while others calmly scoop them up with a napkin? While behaviour is influenced by environment and experience, subtle genetic differences may also play a role. Sometimes the script changes the stage directions. Mutations can also arise from outside sources. Ultraviolet light from the sun, radiation, certain chemicals, and even some viruses can damage DNA and introduce changes. That's why excessive sun exposure can lead to skin cancer. UV rays can disrupt the DNA in skin cells, and if those cells begin dividing uncontrollably, a tumour may form. Fortunately, your body has a team of tiny repair workers on constant duty. DNA repair enzymes patrol your genome, scanning for errors and fixing them before they cause trouble. It's like having an in-house editor reviewing every draft you write. Sometimes they miss something, but more often than not they catch the mistake before it spreads. Still, as we age, the wear and tear on our DNA adds up. Cells accumulate small mutations over time, and although most are harmless, some can nudge a cell toward dysfunction or disease. That's why genetic changes play a role in aging, and why cancer becomes more common in later life. The script begins to fray at the edges, the punctuation becomes unpredictable. But even in this, there's a kind of quiet poetry. Mutations, while imperfect, are also the reason we're here. Without them, life would be static, unchanging, dull. Every feathered dinosaur, every striped zebra, every human eye colour, each one the result of countless mutations, most of them unnoticed, some of them game-changing. Your own genetic code is no exception. You carry mutations that are unique to you, tiny edits that make your DNA slightly different from that of your parents, your siblings, even your identical twin, if you happen to have one. These changes don't make you less perfect, they make you you, one of a kind, even at the molecular level. So tonight, as you rest, picture your DNA not as a flawless machine, but as a living manuscript. A story written in 4 letters, copied billions of times with astonishing care, but never quite the same twice. It is in those gentle errors, those tiny beautiful mistakes, that life finds new directions. Even perfection, it turns out, needs a few typos to keep things interesting. Your DNA may be yours, but it's not just from you. It's a gift, a hand-me-down, passed along through countless generations like an old family recipe written in code. Half of your DNA comes from your mother, and half from your father. And within those double helices lies the quiet legacy of everyone who came before you. Whispers from your grandparents, great-grandparents, and ancestors you'll never know by name, but who left traces of themselves folded into every cell of your body. Let's take a slow stroll through how this works. Humans have 23 pairs of chromosomes for a total of 46. Of each pair, one chromosome comes from your mother and the other comes from your father. That means for every gene you carry, you have 2 copies, one from each parent. These copies may be identical or they may be slightly different versions, called alleles. Some traits are dominant, meaning you only need one copy of the allele for the trait to be expressed. Others are recessive, meaning the trait only appears if both copies are the same. A classic example is eye colour. The allele for brown eyes is generally dominant, while the allele for blue eyes is recessive. So if you inherit one brown eye allele and one blue eye allele, you're likely to have brown eyes. But it's rarely quite that simple. Eye colour, like many traits, is influenced by multiple genes interacting in ways genetic scientists are still working to fully understand. So while we often use simplified examples like brown versus blue, the real picture is a bit more like mixing paints on a palette than flipping a binary switch. The combinations are nearly endless. During a process called meiosis, your parents' cells each mix up their own chromosomes to create sperm or egg cells. Each one of these reproductive cells carries a random selection of just 23 chromosomes, one from each pair. When egg and sperm meet, the two sets come together, restoring the full count of 46 chromosomes and creating a brand new combination of traits. That's why you might have your father's nose but your mother's dimples, or your grandmother's stubborn chin and your great-uncle's unusually loud sneeze. Genes travel through generations in quiet, surprising ways, sometimes skipping a generation before resurfacing in someone new. It's not just your appearance that's shaped this way. Your metabolism, immune system, blood type, and even aspects of your temperament have genetic influences. But before we go too far down the nature versus nurture rabbit hole, it's worth noting that environment plays a big role too. Your genes set the stage, but your experiences help decide the story. Let's take a short detour into the world of Punnett squares. Those little grids you may remember from school. They're a simple way of predicting inheritance patterns. For example, if both parents carry one dominant and one recessive allele for a trait, each child has a 1 in 4 chance of inheriting 2 dominant alleles, a 1 in 2 chance of getting one of each, and a 1 in 4 chance of getting 2 recessive alleles. But of course, most traits aren't controlled by just one gene. The square is helpful, but real life tends to draw outside the lines. Beyond simple inheritance, there's something called sex-linked traits. These are traits carried on the X or Y chromosomes, the ones that determine biological sex. The X chromosome carries many more genes than the Y, so certain conditions are more likely to affect people with only one X chromosome, usually males. For instance, colour blindness and haemophilia are often inherited this way. If a mother carries a faulty gene on one of her X chromosomes, her sons have a 50% chance of inheriting the condition. Daughters with 2 X chromosomes are more likely to be carriers without showing symptoms. Now, not all your genetic inheritance comes from both parents. There's a special kind of DNA found outside the nucleus, in the mitochondria, the tiny power plants of your cells. Mitochondrial DNA is passed down almost exclusively through the mother. So no matter how many generations you trait, your mitochondrial DNA comes directly from your maternal line, passed like a silent message through the ages. This has been incredibly useful for scientists studying ancestry. Mitochondrial DNA changes very slowly over time, allowing researchers to build family trees that stretch back tens of thousands of years. In fact, by analysing patterns in mitochondrial DNA from people all over the world, scientists have traced humanity's origins back to Africa and identified lineages known as haplogroups that reflect ancient migration patterns. So while your genes are a product of your immediate parents, they're also echoes of far older stories. You may carry tiny fragments of Neanderthal DNA, inherited from ancient interbreeding between early Homo sapiens and our now extinct cousins. These bits don't necessarily give you caveman instincts, but they may subtly influence your immune system, skin, or even how you process certain foods. There's something deeply comforting about all this. You're a living mosaic of countless people who came before you. Their triumphs, struggles, travels, and adaptations are etched quietly into your chromosomes. Every time you catch a glimpse of your reflection or sneeze in a particular way, you're carrying forward their legacy in small personal ways. Inheritance also explains some of the more puzzling parts of family life, like why your sibling has curly hair while yours is straight, or how your cousin ended up with the only green eyes in the entire family. Or why Aunt Marjorie insists on reminding everyone that your grandfather had that same freckle on his left ear. It's a game of probabilities, of chance, of molecules aligning just so. Each child is a fresh reshuffling of the genetic deck. You and your siblings may share a lot of DNA, but unless you're identical twins, your combination is unique. On average, you share about 50% of your DNA with each of your parents, and also about 50% with your siblings. but that shared 50% can overlap in different places, which is why you can look alike or not at all. This delicate, random magic makes each person genetically distinct. You're a new chapter in a very old book, a remix of an ancient song, a fresh bloom from a very old tree. So as you drift off tonight, think of the silent threads weaving through time. Threads from your parents, your grandparents, and from people whose names were never written down. Their DNA lives on in you, not as a burden, but as a gift. A quiet inheritance humming softly in every cell, connecting you to the past, anchoring you in the present, and whispering toward the future. If DNA is the script of life, then epigenetics is the director behind the scenes, deciding which lines are spoken, which ones are whispered, and which are left out entirely. It doesn't change the letters of the script, but it determines how, when, and where those letters are read. And just like a good director, epigenetics can dramatically influence how the final performance turns out. At its core, epigenetics is the study of changes in gene activity that don't involve alterations to the underlying DNA sequence. The word itself comes from the Greek epi, meaning on top of, and that's quite literally what it is, an extra layer of regulation sitting atop your genetic code. Think of it like bookmarks, highlighters, and sticky notes scattered throughout your DNA, guiding the cell on which genes to read and which ones to leave alone. One of the most common epigenetic tools is DNA methylation. This is when a small chemical group called a methyl group is added to a DNA molecule, usually at specific points where a cytosine base is followed by a guanine. When enough of these methyl groups are attached, they act as a sort of do not disturb sign, silencing that part of the genome. Another important tool involves the histones, those spool-like proteins your DNA wraps around. These histones can be modified with various chemical tags, acetyl groups for example, that either loosen the diaphragm you're reading or tighten it to restrict access. It's a bit like adjusting the drawstring on a hoodie. Open it up and the cell can peek inside, tighten it and everything stays hidden. But what makes epigenetics so fascinating is that it responds to your environment. Unlike the genetic code you're born with, your epigenome is dynamic. It can change based on your diet, stress levels, sleep habits, exposure to toxins, and even social interactions. It's how your body adapts subtly and silently to the world around you. Take identical twins, for instance. They have nearly identical DNA sequences, yet as they age, they often develop differences in appearance, behavior, health, and more. Much of this can be traced back to epigenetic changes. While their genes remain the same, the way those genes are expressed can diverge based on the unique experiences each twin has throughout life. Even more astonishing, some epigenetic changes may be passed down through generations. While most of your epigenetic markers are reset when sperm and egg meet, some seem to escape the great genetic reboot. Studies in animals and humans suggest that trauma, malnutrition, and even stress can leave marks on the epigenome that affect not just the individual but their children, and possibly even their grandchildren. One famous study from the Netherlands looked at people conceived during the Dutch hunger winter of 1944, a period of extreme famine near the end of World War II. Decades later, researchers found that those individuals had different patterns of DNA methylation in genes related to growth and metabolism, suggesting their bodies had been epigenetically programmed in the womb to expect a life of scarcity. Of course, the field of epigenetics is still young, and there's much we don't yet understand. Scientists are still unravelling the exact mechanisms behind these changes and trying to distinguish cause from effect. But what's clear is that our genes are not static blueprints. They are more like interactive scripts, flexible, responsive, and influenced by how we live. This has profound implications for health. Epigenetic changes have been linked to a range of conditions, including cancer, diabetes, heart disease, and mental health disorders. In some cancers, for example, genes that normally suppress tumours become silenced through DNA methylation, allowing the disease to take hold. But there's good news too. Because epigenetic marks can change, they may also be reversed. This opens up exciting possibilities for new therapies that don't just target the symptoms of disease, but actually retrain the genome on how to behave. Scientists are already experimenting with drugs that can add or remove methyl groups, restore proper gene expression, and potentially halt or even reverse certain illnesses. It also means the choices we make, what we eat, how much we sleep, whether we exercise, and how we manage stress, may have a bigger impact than we once thought. They don't just shape our health today, they may influence which genes are expressed tomorrow, next week, or even in our children. Consider food, for example. Nutrients like folate, vitamin B12, and choline are involved in methylation, helping to shape the epigenome. A well-balanced diet can support healthy gene expression, while deficiencies may throw off that delicate balance. Likewise, chronic stress may alter gene expression in parts of the brain related to mood and memory, while mindfulness practices have been shown to produce epigenetic changes linked to reduced inflammation and better immune function. It's not about perfection. No one can control every epigenetic switch, and life is far too complex to be reduced to a checklist of good habits. But it's comforting to know that your body is listening, adjusting, responding, not just on the surface, but deep down in the language of your very cells. Epigenetics also plays a key role during development. When you were just a tiny embryo, every cell contained the same DNA. But somehow those cells differentiated. Some became neurons, others became muscle, others turned into skin. Now, through epigenetic regulation, certain genes were turned on, others were silenced, guiding each cell toward its unique role in the body. And once a cell takes on its identity, that epigenetic profile is largely preserved. Your skin cells won't suddenly decide to grow into heart tissue, even though they carry the exact same genetic code. The epigenome keeps them on script, maintaining the symphony of your body's functions with graceful coordination. So tonight, as you sink deeper into rest, imagine the tiny chemical bookmarks scattered across your DNA. Picture them adjusting gently in response to your thoughts, your feelings, your memories. Imagine them responding to laughter, to sunlight, to silence. Imagine your cells as attentive listeners, taking notes from your environment and weaving them into the language of life. You're not just a product of your genes. You're a conversation between code and context, between inheritance and experience. You're the sum of countless interactions, some written into you long ago, others shaped by the simple act of living. And within that quiet dialogue lies a kind of hope that who you are is not fixed in stone but written in pencil, with room for edits, erasures, and new beginnings. Imagine you could rewind time, not just a few years, but thousands. Imagine tracing your genetic steps backward, across continents and centuries, through mountains, deserts, and oceans. Hidden within your DNA are clues, little molecular breadcrumbs, that tell the story of where your ancestors once walked, who they lived among, and how the great migrations of humanity shaped the person you are today. This is the world of genetic ancestry, a gentle blend of biology and history. Unlike the branching family tree pinned to your living room wall, this one isn't built from names and birth certificates. It's built from patterns of molecules that have been passed down generation after generation, whispering a record of human movement and adaptation. And it all begins with the idea that while we are each unique, we also carry pieces of a much older, shared story. One of the first things scientists look at when studying ancestry is something called haplogroups. These are groups of similar DNA sequences that share a common ancestor. You can think of a haplogroup as a large family branch that connects people who, many generations ago, descended from the same individual. There are two key types of DNA that are most often used to trace these lineages: mitochondrial DNA, which is passed down from mothers to their children, and Y chromosome D, which is passed from fathers to sons. Since these DNA types don't get shuffled with every new generation the way the rest of your genome does, they're like clean, straight threads running through time. Your mitochondrial DNA, inherited from your mother, changes very slowly over generations, That means it can be used to trace your maternal lineage back tens of thousands of years. In fact, researchers have used mitochondrial DNA to identify what they call mitochondrial Eve, not the first woman, but the most recent common ancestor of all people alive today through the maternal line. She likely lived in Africa somewhere between 100,000 and 200,000 years ago. Y chromosome DNA tells a similar story for the paternal line. For those who have a Y chromosome, it can be traced back to a common ancestor known as Y chromosomal Adam, who also lived in Africa, though not necessarily at the same time as mitochondrial Eve. These figures are not meant to be interpreted as the only people alive in their time, but rather as the individuals whose specific lineages survive to the present day. As people migrated out of Africa and spread across the globe, small changes, called mutations, accumulated in these lineages. These changes didn't usually affect health or appearance, but they served as markers, like genetic passport stamps, that can now be used to identify where a person's ancestors might have travelled and settled. For instance, one haplogroup might be most common in people with Northern European ancestry, while another is more common among people from the Pacific Islands. By comparing your own DNA to global databases, geneticists can identify where your ancestral lines likely originated and how they may have moved over time. In addition to these lineage-based clues, there's also the broader picture provided by autosomal DNA, the kind you inherit from all of your ancestors, not just your direct maternal or paternal line. This DNA is shuffled with each generation, which makes it trickier to follow over many centuries, but incredibly powerful for more recent ancestry, say the past 5 to 10 generations. This is the kind of analysis often used in consumer DNA tests. They compare your genome to reference populations from around the world and estimate what percentage of your ancestry might come from different regions. You might learn that you're 60% European, 20% East Asian, and 10% West African, for example. These percentages are based on statistical matches and are often presented with confidence ranges, since genetics is more of a probability game than a precise ruler. And while these tests can be fun and illuminating, it's important to approach them with a sense of curiosity rather than certainty. National borders are recent inventions, but human genetics stretch back far beyond them. Your DNA doesn't map neatly to countries. It maps to migrations, trade routes, and ancient shared histories. You might also be surprised by what your DNA reveals. Perhaps your family always thought they were 100% Italian, but your results show a trace of the Middle Eastern or North African ancestry. That might reflect ancient trade, conquest, or even just the simple fact that people have always moved, always mingled, always fallen in love across boundaries. There are even signs in your DNA that you carry fragments of other human species. Around 40,000 years ago, early Homo sapiens encountered and interbred with Neanderthals in Europe and Western Asia. Most people of non-African ancestry today carry between 1 and 2% Neanderthal DNA. That's right, your ancient cousins still echo in your immune system, your hair texture, maybe even your sleep patterns. In some parts of Asia, people also carry DNA from the Denisovans, another extinct group of humans. These small genetic traces don't make us any less human. They enrich our story, reminding us that we are not the result of a single neat lineage, but of many ancient crossings and combinations. Our genomes are like patchwork quilts, stitched together from threads spun on many different looms. And each of us carries a unique blend of genetic material that reflects not just our parents and grandparents, but also the migrations, struggles, and discoveries of people long gone. Ancestry through DNA also reminds us of how connected we are. The further back you go, the more your family tree begins to overlap with others. Go back just 10 generations, about 250 years, and you have over 1,000 ancestors. Go back 20 generations, and the number is over 1 million. Eventually, math and migration bring us to an astonishing realization. All humans are related. Somewhere in the misty past we share common roots, so tonight, as you drift into sleep, picture those roots stretching back through time. Imagine each of your ancestors as a candle flickering in the dark, some known, many unknown, all of them lighting the path that led to you. They crossed mountains, built fires, told stories, harvested rice, wove baskets, sailed oceans, and survived winters. Their laughter and worries, their hopes and habits, are tucked into your molecules, carried forward without a sound. And now here you are, unique, modern, singular, and yet composed of pieces that once stood in 1,000 places, a living map of ancient footsteps, a gentle echo of millions, a story written not in ink, but in the spiral of your cells. In the early days of genetics, DNA was like a locked library. Scientists knew it held important information, but no one had the key to read it all. They could study small sections, one gene at a time, but the full story, the entire human genome, remained a mystery. That began to change in the late 1980s, when an ambitious idea took shape: to map and sequence the entire human genome, letter by letter, from beginning to end. This extraordinary scientific adventure became known as the Human Genome Project. It was officially launched in 1990, with the goal of identifying all the genes in human DNA and determining the complete sequence of the 3 billion base pairs that make up our genome. It was, in many ways, a moon landing of biology, massive, collaborative, and quietly revolutionary. At the time, this was no small task. Sequencing just one gene could take weeks. The technology was slow, expensive, and delicate. But with funding from governments and institutions around the world and the collaboration of thousands of scientists, progress began. Machines grew faster, techniques became more refined, and year by year the letters of the genome were slowly revealed. After 13 years of work, in 2003 the project was declared complete. The first full draft of the human genome had been written out, over 3 billion DNA letters, stretching from end to end. And though it was called a complete genome, in truth, it was only about 92% of the total. Some of the most repetitive and difficult to sequence regions remained unsolved at the time. Still, it was a breathtaking achievement, admit. For the first time in history, we had a map of what it means, the most basic molecular level, to be human. We could see where genes were located, what proteins they coded for, and how genetic differences might relate to health and disease. One of the earliest surprises to come from the Human Genome Project was just how few genes we actually have. Scientists had expected to find somewhere between 50,000 and 100,000 genes. Instead, they found only about 20,000 to 25,000, fewer than a grape, and not much more than a worm. It turns out that the complexity of human biology doesn't come from the number of genes, but from how they are used, how they interact, how they're regulated, and how they respond to the world around us. The Human Genome Project also made it clear that humans are remarkably similar to one another. Over 99.9% of your DNA is identical to that of any other person on the planet. It's in that remaining 0.1% where the fascinating differences lie, the variations that shape our appearance, influence our health, and connect us to our ancestral past. These small variations, called single nucleotide polymorphisms, or SNPs for short, are like tiny typos in the genome. Most are harmless, some are helpful, a few are associated with increased risk for certain conditions. By cataloguing these variations, scientists have been able to develop new diagnostic tools, uncover the genetic basis for diseases, and begin to personalize medicine based on an individual's genetic profile. The Human Genome Project also opened the door to new kinds of research. Fields like genomics, transcriptomics, proteomics, and epigenetics have all expanded rapidly in the years since. It's a bit like building a telescope and then realizing you can now see countless galaxies that were once invisible. But beyond the science, the project also raised new questions: ethical, legal, and philosophical. If we can predict disease based on genetics, who should have access to that information? Should your employer know, your insurance company? How do we ensure that genetic data is used fairly and not as a tool for discrimination? These are questions society is still wrestling with. and yet the benefits of genomic research continue to grow. In 2020, scientists announced the first complete gap-free human genome sequence, including the difficult-to-map regions that had been left out of the original draft. And today, it's possible to sequence an entire human genome in just a few days for a fraction of the cost it once required. This ability to read the genome quickly and cheaply has led to remarkable advances in medicine. Doctors can now use genetic testing to diagnose rare diseases, choose the best cancer treatments, and even identify how a person might respond to certain medications. This approach, known as precision medicine, is still in its early days, but it holds enormous promise. The Human Genome Project has also given rise to initiatives like the 1000 Genomes Project, the UK Biobank, and All of Us, a research program aiming to build one of the most diverse health databases in the world. These projects don't just focus on the genome, but on how genetics, lifestyle, and environment work together to shape health outcomes. And while the project was about mapping the human genome, its impact goes far beyond our own species. The same tools and techniques have been used to sequence the genomes of other animals, plants, and even ancient humans. Thanks to these efforts, we've learned more about evolution, disease, and biodiversity than ever before. We've also begun to better understand our own genetic quirks. Why do some people carry mutations that protect them from HIV? Why can some populations survive at high altitudes while others cannot? Why do some people seem more resistant to certain cancers or infection? The answers lie not in any single gene, but in the vast complex symphony of the genome, notes and rhythms playing out in harmony and dissonance. It's humbling to consider that your DNA, the same DNA quietly folded inside every cell, contains information that scientists spent over a decade and billions of dollars to uncover, and yet your body reads and uses it constantly, effortlessly, in the background of your daily life. So tonight, as you settle into stillness, think of the 3 billion letters in your genetic code. Think of the decades of work that went into deciphering them, the long nights in the lab, the careful sequencing, the shared vision of understanding what makes us human, and perhaps take a quiet moment to marvel at the fact that your cells have always known the story. Long before scientists ever mapped it, your DNA was already reading, already building, already composing the intricate melody of you.
You are not just the product of a genome, you are the living expression of it. A message written billions of years ago, still unfolding, still being read with every breath you take.
And so here we are, at the end of our gentle journey through the quiet script that makes you who you are. From the tiny molecular letters A, T, C, and G that spell out your biological story, to the elegant spiral staircase of the double helix, the complex choreography of gene expression, the subtle art of epigenetics, and the ancient paths of your ancestors. Your DNA is more than a code. It is a living, breathing manuscript, still being written, edited, and read with every passing moment. What's perhaps most magical is that your body has known all of this all along. Long before scientists mapped the genome, before the double helix was discovered, before the words gene or chromosome were ever spoken, your DNA was quietly at work, building, repairing, remembering. It doesn't ask for recognition. It simply does what it's done for millions of years, keeps the lights on, gently guiding your cells in their unending symphony of life. And in that soft molecular music lies your uniqueness. You are the only person on Earth with your exact combination of genes, shaped by inheritance, tweaked by experience, gently nudged by time. You're at once entirely yourself, and also a mosaic of every ancestor who came before you. A fleeting moment in a long and beautiful lineage. So as we close the cover on this chapter tonight, I hope you feel a little more connected to yourself, to the people who passed their stories to you, and to the quiet, extraordinary machinery humming inside every cell. Before we part ways, let me ask again, as I did at the beginning, where in the world are you listening from tonight? Is it just past bedtime where you are? Maybe the city outside your window is winding down, lights flickering out, retreats growing still. Or perhaps you're watching from somewhere already deep into the night, where only the hush of the world remains. Wherever you are, I hope you're wrapped in calm, your thoughts slow, your breath steady. And if you've enjoyed drifting through the science of your very own story, I'd be so grateful if you'd give this video a like, or perhaps even subscribe. It helps the Sleepy Scientist continue sharing these slow, thoughtful explorations with others who might need a moment of rest and wonder too. Thank you for joining me tonight. Sleep well, and may your dreams carry you gently through the quiet poetry written deep within you.